folks. Welcome along to the Apologetics Academy, a weekly interactive apologetics webinar where I bring on different scholars, scientists, thinkers, philosophers, theologians from across the theological and philosophical spectrum to present to us on topics of interest to Christians, in particular on those topics that pertain to the question of whether or not Christianity is true. We also cross-examine and critique, evaluate other worldviews as well. This week, we are going to be discussing the dependency graph of life with our special guest, Dr. Winston Ewert, who has done work with Discovery Institute and the Biologic Institute, and with the Evolutionary Informatics Lab based at Baylor University with Bob Marks. There's a number of ways that you can participate in today's program. You can simply watch and listen anonymously. You can also type chat in the chat window to interact there. You can also click the Q&A button at the top of your screen to submit questions in textual form anonymously if you wish. And you can also click the raise hand button when we get to the Q&A. I will promote you to be a co-panelist with the speaker, and you can interact and dialogue that way also. So as I said, we're discussing with Dr. Winston Ewert this week, his paper that was published in the journal Biocomplexity, titled The Dependency Graph of Life, and he's going to be offering us an alternative scientific model to common ancestry to explain the hierarchical classification of life. So without further ado, I'll just turn over to Dr. Ewert. Welcome along.
All right, thanks, Jonathan, and thank you all for coming. I want to apologize a little bit in advance. I'm unfortunately at the tail end of a cold, which mostly means I will occasionally go into coughing fits. So I apologize for when that happens. All right, the first thing I want to talk about today is who I am and what my background is. I am first and foremost a computer nerd, and I have been for a long time and probably always will be. Very interested in computers and technology and that side of thing, and we'll note that is sort of a theme for everything else that I've ended up doing. What happened is I read the book The Blind Watchmaker by Richard Dawkins, in which he has a computer simulation of evolution, which involves the phrase, "me thinks it is like a weasel." That led me to getting interested in the subject of digital evolution. And through that, I ended up being down at Baylor, working with William Dembski and Robert Marks, and I got a PhD there and wrote a number of papers relating to the subject of digital evolution, or computer simulations evolution. I also did work on algorithmic specified complexity, which is applying some ideas from algorithm information theory, which is a part of theoretical computer science, to Dembski's work on specified complexity. And I have a few papers on that as well. And so you'll note, as the theme is, as I've sort of alluded to, I look at things from a very computer-oriented perspective and figure that there's no problem that can't be solved with a proper application of ideas from computer science. And that's going to continue somewhat, but there's a bit of a shift in what I'm talking about now from my previous work. And in particular, my previous work has focused on complexity. And this is common to most people in the intelligent design side of things. They like to focus on things like specified complexity and irreducible complexity, because what the argument is, and I think it's very strong, that intelligent design is the only theory that has a halfway plausible account of the complexity of life. There's not really any other way to explain the functional complexities we find in life other than an intelligent designer. Our critics, however, would prefer to focus not on the complexity of life, but the homology of life. They want to focus on the similarities and the patterns of similarities between different living things, because they think that intelligent design doesn't have a good answer here, and that the patterns we see are nicely explained by evolutionary theory and by common descent in particular. However, it's not as if we can't explain homologies. If we look at designed artifacts, we see homologies all over the place, and I have various pictures here to depict examples. So we have, for example, on the left, car gauges on a car dashboard, and you'll note that any two cars you look at have pretty much the same gauges. You'll find they're typically in a similar order. If you look at circuit boards, as in the center case, you'll find they've got similar colors, and they've got similar components welded onto the circuit board. So we have what we would think of as homology as another designed artifact. And on the right, we have the two smartphones, an iPhone and the Android. And they're similar enough to the point that it's difficult to tell at a glance which is the iPhone and which is the Android. So there's not really a problem at all in terms of explaining homology per se in a design framework. In fact, if we didn't find these similarities in life, it would really be an argument against design because it would be completely different than our experience of how designers operate. However, the challenge to design really comes from the fact that there seems to be a pattern in how different species share similarities, and this pattern is usually identified as a nested hierarchy. The idea being that different life forms can be divided into groups, undergroups, undergroups. So if you look at, for example, all the animals, they're divided into the vertebrates and invertebrates, and you can further divide vertebrates into tetrapods and other things, and tetrapods into things like mammals, birds, and reptiles, and you can keep on dividing each group into subgroups until eventually you come to the species that we know today. And so this has long been the standard way of classifying life, and common descent gives a fairly nice and simple explanation for why life would seem to nicely fit this hierarchy. Each group here corresponds to some long gone species and all of their descendants. So there was some long ago species that is the common ancestor of all our current animals, and all the commonality that we find across the animal kingdom all derives because those features were present in the first animal, and they've simply been inherited by all animals since then. And we have tetrapods, for example. There are some original tetrapod species, and all tetrapods are descendants of that original species, but they also inherited similarities from animals because that first tetrapod was also a descendant of the first animal. Now, a question that is sometimes argued about is whether or not life does fit a nested hierarchy, because you can point to many cases that are somewhat exceptions, that things that don't seem to fit the hierarchy correctly. And indeed, whole portions of life, for example, bacteria, are very difficult to fit into any sensible hierarchy at all. But I think really that's the wrong question to ask, because the truth is life and a hierarchy is really not a binary question. It's a question of a spectrum. Life resembles a hierarchy to some degree, and we could think of different ways of measuring it, but if we could quantify it, it could range somewhere from not fitting a hierarchy at all to being a perfect fit to a hierarchy. So no fit of a hierarchy is, every similarity is randomly distributed and there's no pattern at all. And a perfect fit would be a case where every character fits the tree exactly with no exceptions. And life falls somewhere into here. Now, people will argue exactly where I should put life in this case. I could put it somewhere in the middle here, and some people would argue, well, it should be closer to the left, that there's lots and lots of data that doesn't fit, and there's just slight resemblances to the hierarchy. Some people would argue, but others would argue just as strenuously that actually it should be much closer to the right, that, well, yeah, there are exceptions to the hierarchy and things that don't seem to quite fit. Mostly the data pretty much fits a hierarchy. And so there's a lot of argument you could put into that, and it really depends which data you're looking at, how you're measuring it, which species you're considering. But I think what I could assert is we're not really close to the no fit at all case. We're not close enough to no fit at all that we could say, well, there isn't anything to this. There's clearly something to the fact that life is classified as a hierarchy, and there's something that needs to be explained. But it's also not really close to the perfect fit case, because there's enough data that doesn't fit the hierarchy that it doesn't make sense to just say, well, it's a hierarchy and that's the end of the story. Now, your typical argument for common descent works something like this. The theory is that everything under the blue half rectangle is best explained by separate ancestry, and everything under the red half rectangle is best explained by common descent. The argument is basically, well, either you have separate ancestry or common ancestry, and since separate ancestry really only explains things that have either no fit to a hierarchy or almost no fit to a hierarchy, just enough that it could just be a random chance event that there's some resemblance to a hierarchy. Everything else is best explained by common descent. And they say, well, even if life isn't all that close to a perfect fit, it's still sorry, my train list. Even if it's not a perfect fit, it's still closer to being a perfect fit than no fit at all, effectively. The critics of common descent think it's not really fair to claim basically the whole spectrum of possibility as evidence for your theory, and you should really only infer common descent if data was much closer to a perfect fit than it is. And whatever the merit of that, it raises a question, well, what exactly is the explanation for data that falls in this range? Because we can't just say it's random chance, because that would sort of be the blue box, and we're clearly too far away from a random chance distribution of characters to invoke that explanation. Even if you could argue maybe we're not close enough to common descent to invoke that explanation, but this leaves the question of what exactly is the explanation. And I think from the common design side of things, there hasn't really been, up to this point in time, a really good model to explain what we're seeing. And this brings us to today, where I've put forward my model. And as I alluded to earlier, I look at everything from the perspective as a computer scientist and figure there's no problem that can't be solved by thinking about it like a computer scientist. And so I look at this from the perspective of how would a software engineer design all these various species. And for that, I want to start by talking about how a software engineer creates computer programs. So when a software engineer creates a computer program, they do not create computer programs from scratch because effectively we are far too lazy. What we do instead is we have a repository of modules. So each module has some particular task that it performs. That task could be downloading a file off the internet, spell checking a document, comparing how similar two pieces of text are, any number of things. And for a wide variety of different things you might want to do in a program, people have created modules that do it. What the programmer will do is he will identify the modules that provide functionality that he needs, and he will copy those into his program. Right? And so literally it's effectively copy and paste, but automated by the computer. Now, invariably you'll find when you write a program that there's some functionality you need that no one has created before, and so you'll create some modules just specifically for your one program, and that will round out all of the modules in your program. So the first assumption of my model is that this is in fact how biological species were designed. That biological species pull from a selection of modules already existing in a repository, and those are put together to form the actual species. And each module forms a small piece of sort of the overall blueprint of the species. And so when we find homologies between different species at whatever level that is, it's because the species share modules. And the modules end up exhibiting themselves as homologies in different species. Now, I am by far not the first person to come up with this idea of modules, or the idea that a designer would reuse designs between multiple species. So that's long-standing argument being made by people who are holding to a common design, and it's really, to some degree, what the idea of the name common design refers to. What I've introduced that's novel is the idea of a dependency. And to explain this, I want to talk about how a software engineer would think about designing his house. So effectively, if you want to design your program, you're defining a module that defines your program. In this case, I'm going to think I want to define my home, so I want to define a module that defines my home. But I'm not going to start from scratch. I'm going to say, well, what modules do I need in order to define my home? And I'm going to say, well, I'm going to need like a home module, we'll say, and my house happens to have an office in it, so I'll have an office module. And the way it works is that effectively I say my home, it's a home plus an office plus these other modifications specific to my home. Effectively, think about common descent being descent with modification. The way that a software engineering builds things is effectively dependency with modification. And we call these other modules that we're using dependencies. And what we specifically say, we say that my home module depends on the home module and it depends on the office module. But we can actually go further than this because we can look at, well, how is the home module defined itself? And we note, well, the home module itself would be defined in terms of other modules. For example, an occupied building module, which effectively a building people would actually spend time in as opposed to a more generic concept of building. You also have important things that any home is going to have. You better have a bathroom in your home and a kitchen and a bedroom. If you're missing any of those things, what you have is probably not actually a home. But that each of these could be further developed. You could talk about, occupied building is defined in terms of a more generic building module, as well as things like a washroom. Any building you're going to have people spend time in, it's pretty important that it does in fact have a washroom. And of course, we could define building in terms of even more generic concepts. Then if we look at things like a bathroom is really a special case of washroom. So while a washroom is something you would have in any building, your bathroom in your house is going to have additional things like a shower that you wouldn't find in a generic washroom. Generic washroom we would define in terms of an occupied room, and it has important things like sinks and toilets in them. And we could further develop this with different kinds of rooms being, various special cases of this occupied room. So those are all different modules that would depend on the occupied room module. And then the occupied room module itself would depend on other things that you need to have in those cases. And we could fill out the various different things we would find in different sorts of rooms. But then we could also look even at sort of the services these things require, such as you need hot water and cold water and things like hot water tanks to support that, and all the various different entities and modules and concepts that you need to include into your final house design in order to have a complete house. Now, this is how software is built. Every module defines a list of the other modules it depends on. And when you hand your program over to the computer to produce the final result, it traces through all of those modules. In many cases, actually going to the internet and downloading them into your computer in order to put them all together to produce the final product. And what we do is we call this collection of modules and dependencies a dependency graph. So all the various ovals here are modules, and the arrows between them are the dependencies, and together the whole thing is called a dependency graph. And the thing to note here is that if you have another home someone defines, they would pull in some of the same modules as my home. So for example, any other home is probably also going to pull in a home module, and is thus actually going to depend on many of the same modules as my home is. And thus you'll get a lot of similarities between my home and anyone else's home because we're using the same module. And even if you don't depend on the home module itself, if you depend on, say, a kitchen module, maybe it's a restaurant or something, you're still actually going to exhibit a lot of similarities to my home because we're actually going to pull in the kitchen module and everything the kitchen module depends on. And so the fundamental idea of my model here is that this is in fact how biological species are defined, that you have modules and the modules have to follow this dependency graph. That means you can't just have any arbitrary collection of modules in a species. It has to follow a particular pattern that follows a dependency graph, and that is what I think produces the nested hierarchy pattern. Now, how does that work? Well, let's take a look at a small part of the tree of life. So here I have a selection of mammalian species. Now remember that the common descent story here is that long ago there's the common ancestor of all mammals, and that's split into the different categories we have, again splitting multiple times in most cases to produce the various different extant species. And what I propose is that all we need to do is turn the arrows in the opposite direction. And now, instead of this being a tree of life, this is a dependency graph. So Mammalia is not actually a common ancestor of all the animals. Instead, Mammalia is a module. And all different mammals exhibit a lot of similarities because they all depend on the Mammalia module. And if you look at other cases like the Carnivora module, it also depends on the Mammalia module, which is why you don't have any Carnivora which aren't mammals. It's because you can't make that because it would not follow the dependency graph. Carnivora depends on Mammalia. And this is what explains sort of the nesting effect that we're seeing that is interpreted as evidence for common descent is actually a reflection of the dependency graph. There is, however, a problem. This doesn't actually look quite like a real dependency graph because each module has only one dependency, and that would be very atypical of a dependency graph. Typically, a module has multiple dependencies. In order for this to look like a dependency graph, there should be more modules besides the ones that we're seeing here. So it should look something more like this. So if there's a marine module, which could explain similarities between different marine animals or an echolocation module with similarities between different echolocating species, this would actually look realistically like a dependency graph. The idea of my hypothesis is that we've understood it as a tree because, well, we were looking for a tree because that's what we thought it was, and we've identified fairly large modules or the set of modules which fits a tree which best approximates the overall dependency graph. And it happens that some modules are quite large and they dominate any sort of measurement of similarity. And so that's relatively easy to get to just focus on those modules and restrict yourself to a tree. You get something that looks like a nested hierarchy, and it's an approximation to the whole truth of the dependency graph of life. All right, so this is a picture of myself as a baby. It's actually the first picture of me that was ever taken, and my name is Winston because they thought I looked like Winston Churchill. I'll leave it to you to assess whether or not that was a good assessment. But the reason I have the picture here is because I want to emphasize the fact that my theory here is also a baby. I really, at this point, I've just put it out there because no one else has proposed the idea of a dependency graph before, and it's currently under investigation. I'm not claiming at this point to have established that it's true or that you should all believe that I'm correct. But I think it's an interesting idea worth investigation. So for the purposes of my first paper on the subject, I wanted not necessarily to evaluate, is it true? Because, well, that's a difficult problem, and I think there's going to have to be a lot more work before we can ask the question, it's true. I want to ask a more, less ambitious question, which is, does the data fit a tree or a dependency graph better? Now, note, I didn't say, does the data fit common descent or a dependency graph better? The thing is, common descent is very difficult to test because, well, it has a basic tree structure. That's relatively easy to model. When you start throwing in all the different mechanisms like horizontal gene transfer and gene resurrection and incomplete lineage sorting and all the variations of that, it's very difficult to do any kind of reasonable test on common descent because of that. So want to stick, at least for now, to a simpler question of, does the data look more like a tree or more like a dependency graph as a first evaluation of, is it possible that my model is correct? However, there are a number of difficulties in trying to assess this question. The first difficulty is that you can always fit the data set to a tree. Now, this has often been pointed out. If you take a bunch of characters, no matter what kind of characters there are, and you feed them into a tree building program, it will always give you a tree, right? There's no distribution of characters that would falsify a tree structure, and that's unfortunate. The same is true of the dependency graph. There are no characters which you can't also fit and give a dependency graph story to. So ideally, we would have a case where there'd be some distributions which would be prohibited by the theory, and then we'd go out and look for those distributions, and we could have a nice, clean falsification of either model. Unfortunately, that's not the case we're in. So we have to ask instead the question of which model fits the data better. And we do this typically by looking at it in a probabilistic fashion, because we know common descent would say certain combinations, certain species should exhibit similarities much more than others. So, for example, chickens and turkeys are a lot of genes and similarities in common because they're closely related birds. But if you look at, say, a zebrafish, they have similar names, but they should be very distantly related. And so they shouldn't really exhibit a whole lot of similarity aside from generic, they're both some similarity because they are, of course, related under common descent, but they shouldn't exhibit strong similarities. But the further problem is that a dependency graph is always going to fit better than a tree. The issue is that, as I've discussed earlier, you can take a tree and convert it into a dependency graph just by reversing those arrows, and then you could almost certainly be able to find a few cases to add modules, and thus better fit the data by finding a case where modules help that. But the problem is that the dependency graph is always going to be more complicated than a tree. Now, you can think of this two ways. One is that you're going to have more modules than you are ancestral species. So basically, any set of species can be connected by a set of common ancestors equal to the number of extant species, but you could have easily, in order of magnitude, more modules than that. And so that's obviously much more complicated. And additionally, If you look at it simply in terms of the number of possible trees and the number of possible dependency graphs, well, the number of trees can be quite large. The number of possible trees that could relate a group of species is dwarfed by the number of possible dependency graphs. And so this raises a problem because obviously we want to prefer theories which fit the data better, but we'd also like to prefer theories which are simpler, that have more parsimony. The question is, how do we trade off these things? Well, the answer to that question is Bayesian analysis, named after the Reverend Thomas Bayes. The effective insight here is that we can't just look at how well the data fits, we also have to consider the priors. And in particular, we have to consider the prior on the dependency graph. So there are many possible dependency graphs that could exist, and we have to assign a probability to each of them. And when we're looking at how well the data fits the dependency graph overall, we have to take into account not only the probability of the dependency graph, but also the probability of not only the probability of data under the dependency graph, but also the probability of the data of the dependency graph itself. And so by taking into that kind of prior, we could, in a principled way, decide whether the data fits better enough dependency graph to think that's the better model, or whether we think it's just a statistical fluke and there isn't anything interesting there. Now, in order to do sort of this Bayesian analysis or Bayesian model selection, I have to make a number of assumptions. One, I'm only going to consider gene family presence or absence. So I don't look at details of what the gene sequence looks like. I just look at whether or not the gene family is present in the species at all. This is primarily just for the first attempt at evaluating this idea. I want the simplest model possible, not to get into unnecessary complications yet. To figure out which gene families are which species, I looked at a number of different gene databases and just said, however you classify the gene families, whichever gene families you tell me are which species, I'm going to take that database's word for it. I didn't do any of my own analysis on which gene families were present in which species. The second point is that this is pairwise model comparison. So we can take two models and tell you which one is preferable. Now this always has the problem of the third model. So the issue is that a third model is some other model besides your two you're testing. And when you're doing Bayesian model selection, it can tell you which of the two models you're selecting is better, but it can't tell you anything about whether there isn't some other model which is even better. Particularly, recall that I'm only looking at a tree model versus a dependency graph model, so maybe some common descent plus a bunch of other mechanisms model could be better. That's not really something that I can evaluate here, and it's not something that I do evaluate here. I'm assuming here that gene families arise only once. So this precludes things like horizontal gene transfer or gene resurrection. So my assumption is a gene family can't have popped into two different independent species. If they did, they would be different enough, they would have ended up getting classified as different gene families. So that's my assumption. Getting the tree of life, I just followed the NCBI hierarchy. Obviously, there's going to be some debate about how exactly species are related, and people might dispute what's in the NCBI hierarchy. But my assumption is that's close enough that we can get some sort of idea of this. In order to create the dependency graph, I start with the tree of life from NCBI, and I convert it into a dependency graph by reversing the direction of all those arrows. And then I systematically test different potential modules to see if adding them helps improve the fit to the data, taking into account that prior, and ran that for a while to see what results I would get. All right, so firstly, what I did with this is I did a couple of sanity checks. So the first are some evolutionary simulations. So I took a program called Evil Simulator, which simulates a process of common descent and gene of evolution. It runs for a while. It outputs a bunch of different species with a bunch of different genes that have undergone evolutionary development. And then I processed that through OrthoFinder to turn them into gene families. And then I ran them through my analysis pipeline to see, does it think that that data looks more like a tree or more like a dependency graph? And you can see the results here. So I did it for 5 different runs with different settings of the Evil Simulator parameters. If we look at the second column, you'll see those numbers with the heading tree versus null. So these are the log Bayes factors. So to understand what this means, positive numbers favor the first model and negative numbers favor the second model. So you see these are the largest positive numbers. That favors the tree model over the null model. And so the tree model, as I discussed, is a simple model of common descent, as it were, and the null model is effectively a star phylogeny. So this data looks a lot more like a tree model than the star phylogeny. To understand the scope of these numbers, they're bits. So that means that effectively 6.6 bits corresponds to your 0.01 p-value-ish thing, or sort of generic decisive result. And so this, of course, is much beyond 6 bits, because each bit doubles sort of the confidence of it. So it would basically be intellectually irresponsible to claim that actually this was produced by the null model, not the tree model. If we look at the third column, we have the dependency graph versus tree. You'll see these are negative numbers, and that says that the data here fits a tree a lot better than a dependency graph. And that's, of course, important because it shows that, well, I do, in fact, correctly identify this as tree, and it doesn't look like a dependency graph. And also, it's not the case that no matter what happens, I would always think it looked like a dependency graph. As a second sanity check, I took software applications, which are produced by following a dependency graph, and asked what happens if I put it through the analysis. Now, of course, software programs don't contain genes, but I extracted what are called functions and figured those are similar-ish to genes and ran them through the same analysis. And we see here, we have tree versus null. It looks a lot more like a hierarchical structure than a star phylogeny. But it looks much more like a dependency graph than it does like the tree. Now, it might be surprising to you that the JavaScript applications would fit the tree, but this is actually a pretty critical point for my theory, because my claim is that life is following a dependency graph, and it's been misinterpreted as a tree. And for that to be plausible, it should really be the case that we could actually do the same thing with things we know are produced by a dependency graph, like software applications. Now, to give you something of an idea of why this happens, I have here the tree of software applications. And this tree was produced by systematically testing all the possible trees effectively to figure out which one best fit the data. Now, you'll notice the colors. What's happened here is I've colored the different modules according to their framework. So most programs are built on a framework. This is effectively a large module that provides your basic building blocks for an application. It'd be roughly analogous to sort of a module defining the body plan for a species. And so, as you might guess, different programs using the same framework, they tend to exhibit a lot of similarities. And you can see that different frameworks tend to be grouped together in parts of the tree. Under an evolutionary interpretation, we'd think something like, well, there is a common ancestor of all programs using a particular framework. Of course, that's not actually what happened. There's actually a module thing going on. The case that you might find a little interesting is the case of the tan programs, which are branching out of the pink programs, despite the fact that frameworks aren't actually related. Interestingly, this is actually trying to explain the case of H and NG2 in React News, because they share a large module outside of a framework, and so they're being put close together in the tree to attempt to accommodate that similarity. So what this shows effectively is that a tree can kind of approximate a dependency graph in various ways by choosing a tree which kind of in some way resembles the truth of the dependency graph. Of course, what we're mostly interested in is what does the biological data look like? So what I did here is I looked at 9 different gene databases, and I took their definitions of gene families and which species the gene families are in. I only looked at metazoans, and I asked, which model does it look more like? So firstly, we could look at the tree versus the null. And as you can see, we have fairly large positive numbers saying that data systematically across all these databases looks a lot more like a tree than it does like a star phylogeny. And this is effectively the test which has been done many times in the literature to make an argument for common descent. Because what they've always done pretty much is they've compared common descent to a null model. Usually they're not even generous enough to make it a star phylogeny, but effectively it looks a whole lot more like a tree versus the null model. But of course you have to remember, whenever you're doing this sort of analysis, the problem of the third model, what models aren't you testing? And of course, in this case, I'm saying, well, here's another model, the dependency graph, and we can ask, is that better? And we could look at those results now. Now, you probably already guessed by the fact that I wrote the paper and got it published and I'm giving this presentation, the dependency graph does in fact fit the data better than a tree. And it does so by at least 10,000 bits. Now, if you recall, the 6.6 bits is considered the decisive level in a sort of generic way, or the p01 p-value. So this is a fairly strong result. Basically, the data absolutely does fit a dependency graph much better than a tree. However, keep in mind, just as the comparing the tree versus the null had the weakness of maybe there's a third model, maybe there's yet another model which could beat out the dependency graph. All I can really say is it looks more like a dependency graph model than it does a tree model. So nextly, I'm going to look at some examples of what sorts of things are being found in the data that tend to favor the dependency graph. Now, I want to have some caveats here because the overall strength of the result is really the fact that I get quantitatively looks more like a tree than a dependency graph across 9 different gene databases with different species and different definitions of gene families. When I look in the upcoming area, I'm going to look at particular examples that come from, say, one database at a time or a small selection of species, and those examples are much more easily explained perhaps in other ways than the overall result. So there could be contamination in databases or problems with the data or other weird things that have happened in evolutionary history that could explain the individual examples. And so I'm showing them as information, but the overall case really is not based on the fact of these examples. Just want to be clear on that point. So the first case I want to look at is the scarlet sea anemone and the Florida lancelet. Now, these are in different phylums, so they should be very, very distantly related. Right? They're as distantly related as they can be and still be considered animals. And yet, according to every gene database I check, they have a lot of genes found only in these pair of species and in no other species. So my model infers that that is explained by them both sharing a module. Here I have a selection of fish species, and take particular note of the species in green. So according to at least the database that I'm looking at here, these species share a bunch of genes not found anywhere else in the animal kingdom. Now, common descent can explain this. So common descent can say, yeah, these gene families were present at the root of all of these species, and they were lost just at 3 different points to end up with this particular distribution of genes. But we don't find that any combination of different losses is equally populated. This particular combination is more populated than others, which looks like actually these species happen to share a module of some sort. And when you say have enough cases that look like this across the whole animal kingdom, that is the sort of thing that tends to end up favoring the dependency graph over the tree. So here we have a dependency graph depicted for the homologene database. So the homologene database only has 13 animal species, and so they're depicted in the blue, as the blue rectangles. And so I can actually depict all of the species because it's a relatively small database. However, I cannot depict all of the modules. So there actually are 145 modules that I inferred to exist from this database. So I'm only showing the relatively large modules here. And the modules are the circles. And you'll note the circles are different colors. So green modules correspond to taxonomic categories. So these are things that more or less common sense says, yeah, there should be a bunch of genes here. But the orange modules are ones that don't correspond to taxonomic categories. So I infer that there seems to be enough genes shared between those combinations of species to infer that there's a module there and they don't simply come from the common ancestor. And so we have things you would expect on common descent, like chimpanzees and humans share a module, mice and rats share a module. But then there's also some interesting surprises. For example, chimpanzees and rats sharing a module, or rats and rhesus monkeys sharing a module, or a module shared between the chickens, frogs, and zebrafish. So this is another database, and I've restricted the species here just to be some relatively familiar primates. And again, we have what we expect under common descent. We have things like there's a large module shared between humans and chimpanzees. But then there's also a number of other modules, even some relatively large modules, of suggesting gene families which aren't really shared according to the distribution expected by common descent, and infer that by inferring these modules, we can actually better explain the distributions of those genes. Here we can move further out and look at some familiar mammal species. And so we have things you'd expect, like dogs and cats share a module, as do the mice and rats. But there's also a module shared between the dog and the rodents, and various things like that. Here I have this. This here, what I did is I just selected 10 species at random and plotted their dependency graph. Again, I have to remove smaller modules and other modules that aren't relevant to these species in order to have something that is remotely comprehensible. But we see here that just across the animal kingdom, we have lots of different modules we're seeing that are inferred by this process. And then these all correspond to genes which are distributed in a way that's not really fitting the common descent story and actually ends up better fitting the modules or dependency graph story. So talk a bit about the question of what has the response been to the paper thus far. So one response has been that it was published in Biocomplexity, therefore we shouldn't pay any attention to it. And if this is your attitude, my attitude is that I don't need to pay any attention to you. So another line is to say that it doesn't address their favorite evidence for common descent. Now, what I find here is that everyone seems to have their own favorite line of evidence for common descent. So some people say, well, the fossil record settles it, and some people say the nested hierarchy settles it, and some people say comparative anatomy settles it. Everyone has their own favorite. And so if you didn't think the nested hierarchy was a particularly interesting line of evidence for common descent, well, yeah, you're not going to be really impressed by the fact that I have a model for it. And ultimately, I think if we're going to have a truly competitive model for common design, yeah, we do need to address all the lines of evidence that are presented for common descent. In this paper, of course, I'm just focusing on the nested hierarchy. A common objection I've gotten is that it doesn't address sequence level similarity. So I just looked at whether the gene family was present or not present. I didn't look at the details of the sequence. And I think a lot of cases, this is simply an attempt to dismiss what the paper shows, and it doesn't really work because, I mean, the fact that whether or not the gene family is present or not is effectively a sequence level similarity is just at a high level of granularity. So that's not going to disappear if we get down to the sequence level. At the same time, yeah, there's details and features that happen at the sequence level which are simply skipped over by the analysis I've done so far. And ultimately, the model needs to be developed further to be able to address the sequence level question. And if you want to just say, oh, I don't think you can do it, well, just watch me, we'll see. A common objection is also that I don't include the mechanisms known to be in operation. So I tried to make a big point here. I tested whether it looks like a tree, more so than testing whether it looks like common descent, because I don't include things like horizontal gene transfer, incomplete lineage sorting, etc. Now, I did study specifically metazoans to try and minimize this as much as I could. Metazoans aren't thought to include horizontal gene transfer to a significant degree. And because I'm only looking at gene families, incomplete lineage sorting is a much less interesting question in terms of trying to address that. But ultimately, with all these mechanisms, yeah, further research is needed to try and evaluate whether we can test the individual theories that would claim that this or that explanation. But I think a key point to realize here is up to this point, common design has had a challenge to explain why does the data look like a hierarchy. And now that there's a model for that, the question now has to be asked, well, why does the data look like a dependency graph? And so maybe you can come up with some other explanation for why it looks like a dependency graph by invoking some of these other mechanisms. But that's now the question that has to be answered. I think perhaps the most serious objection is that the annotations or a lot of genomes are incomplete. Life is messy, and our data about life even messier. A lot of the gene annotations are incomplete. There are genes which are present in genomes, but they're not present in the annotations. And so the argument is made that maybe what I'm doing here is I'm actually just inferring modules because of missing gene annotations. I don't think so. I mean, further work is necessary to fully evaluate this, but in order for this to be a working explanation, it's not just that the gene annotations can be missing. They have to be missing in a particular way, which happens to look like modules. And I think that's not a terribly plausible account. But I'll grant, for all of these questions, further work is necessary in order to further address them. So where does that leave us now? Firstly, I would say that yes, actually design can explain resemblance to a nested hierarchy. So up to this point, people have tried to make the argument common descent branching process is the only way we can explain resemblance to a hierarchy. So given that there's any resemblance to a hierarchy at all in life, it's got to be common descent. And really what I set out to do with this paper was to take that argument down, and I think I've succeeded. I think that you can no longer make that argument. You've got to actually make at least a much more sophisticated argument if you're going to try and make an argument for common descent off of the nested hierarchy. I think at this point in time, it has not been established whether or not my model is correct. So I think there's, I talked about the various open questions and there's limitations to what I've done this far. So I think honestly, we don't know whether or not I'm right yet. I don't think any of the attempted responses are going to pan out. I'm sure that my critics will disagree with me, and we'll have to see how that happens. But I think at this point, it is still up in the air. But ultimately, I think what's promising here is I'm hoping it's a direction of future potential research. I'm hoping that this brings out a platform and inspiration and motivation for me and others to develop the idea of common design into a comprehensive model that can explain everything that's going on with that seems to be evidence for common ancestry. And I'm hoping that this is really just the start of a comprehensive model in that direction. And if that's all that this paper accomplishes, I will be more than thrilled. So thank you all for listening and listening to my presentation. And yeah.
Well, thanks so much, Winston, for that very interesting presentation. Folks, this is the interactive portion of the program. If you'd like to interact with Dr. Ewert, then click the raise hand button or submit a question by hitting the Q&A button, and I'll be happy to take questions that way as well. Give people a couple minutes, otherwise I've got a few things I can ask off the back of that. So could you go over for me the criteria that you used for determining whether the data better fit the common or the tree-like model or the your alternative model that you're proposing?
The criteria. So
Yeah.
I use a Bayesian model selection, which means that both of the models, I define a probabilistic model that says what's the probability that this gene would be located in this particular set of species. Probably difficult to go in this venue over this precise mathematical definitions there, but it involves things like, well, there's a certain probability the gene was introduced in a species and probabilities of it being lost as it goes down the tree of life and such things. And effectively, I use a very similar model for the tree and the dependency graph, except for the fact that the dependency graph allows multiple modules to come in, and the tree of life, of course, you only have one ancestral species. And then I evaluate which of those ends up handing the higher probability to, well, I sum those, well, I multiply those probabilities across all the different genes in the data set to figure out overall which one has a higher probability for the data, taking into account the prior on the dependency graph. I can't hear you.
Oops, sorry, I was muted. There's a question that was submitted by Kevin Ross, who has a physics background. He says, what is your metric? Did you say one of your metrics was a log probability metric? Have you related your metrics to entropy?
So I use a log Bayes factor, which is a log of probability ratio. So no, I've not related that to entropy. I don't immediately see any way that I could usefully do that.
Mm-hmm. Have you got any ideas for how you could relate this at the sequence level? You mentioned it briefly that your model doesn't cover this at this point. I'm thinking in particular of cases where you obviously have the redundancy in the genetic code, and you can have multiple codons which code for the same amino acid. And so you can change the sequence at the DNA level without necessarily affecting the amino acid sequence level. And so how you would explain kind of the nested hierarchical distribution that's seen at the molecular level, which doesn't seem to affect the function.
Right. So I'm hesitant to go too far into discussing that because I have vague ideas of how to do that, but I haven't really tried to build it out yet. The indication that I've seen is that the speed of transcription and such is affected by which codons you're using. And there seems to be particular biases in the selection of those codons, probably directed at exploiting that, which suggests to me that quite plausibly there is slight functional benefit to particular things. And while I think natural selection would have a difficult time accounting for that because they're such slight, I think design framework will actually have a much easier time accounting for that kind of thing.
Okay. Coelacanth in the Q&A asks, if it's true that major gene families aren't distributed in a tree-like pattern, this seems like a pretty big problem for evolutionary biology. I would have expected that someone in the mainstream community would have discussed this problem in the past. Have any mainstream evolutionary biologists ever observed this fact before and discussed it? If so, who and how did they explain it? If not, why do you think this problem has been missed?
So there are a couple of papers I've seen where they look at it. They seem quite comfortable either saying there was horizontal gene transfer that explains these things jumping from place to place. Some people say, well, we can't do that. These are animals. They don't go with horizontal gene transfer. And instead they're saying, well, genes were just lost at various points in time. So you can explain, oh, these genes not really fitting a pattern by saying, well, yeah, genes are just being lost all the time. And so, yeah, so whole portions of the tree of life are missing their genes in that kind of thing. So that really, I think, doesn't bother them particularly much that their genes are missing that way because gene loss is easy to invoke.
Right. Rob in the Q&A asks, you have presumably used a small subset of all the data out there. Any idea of the fraction of the data you have drawn upon?
Well, so I guess so the largest database I use has 330 species in it. I know that's arguably a tiny fraction of all the species out there. Of course, I don't know that there's that much more which we have good data for at this point. You are muted again.
Sorry. Rob asks, what are you planning to do next?
Thus far, I'm changing my mind every 5 minutes about what to do next, so I can't really give you a useful answer to that question.
Kevin Ross asks, does your dependency model potentially give a better way to describe or quantitatively assess fitness and possibly show that proposed lineages either do or don't work?
Fitness. So I don't immediately have any thoughts of any connection between what I've done in fitness and evaluating that kind of question. I guess maybe you're thinking something along the lines of, well, if this gene used this gene, we could say this lineage doesn't make any sense because the gene it required wasn't there, and you could do something with that, might be what you're thinking. I haven't really thought along those lines.
Currently no submitted questions, so give folks a few minutes if they want to ask anything else. We have a question from Rob. Are you releasing your code into the wild?
I've released the data that it has, like the particular dependency graph I inferred and a script that evaluates the performance of it. I haven't at this point released the dependency graph inference algorithm because I didn't want people getting distracted by any quirks in that when evaluating what I'm doing. I'm hoping at some point to produce a version that I would be happy to let people work with and do stuff with, but I haven't at this point.
Everyone seems convinced.
Convinced, or I was really confusing. I'm hoping everyone's convinced.
Yes.
And the people who couldn't be convinced either couldn't make it or thought apologetics was a waste of time.
Yes. There are a few. There are several people with science backgrounds in the audience, so that's good. Rob says, has there been any scientific response from others?
This depends how generous you want to be with the label scientific. So what I've seen thus far, I've seen it discussed in forum threads and blog comment threads. And my general attitude is nothing useful happens in a forum thread or a blog comment thread. So I talked about some of those responses I've seen. Some of them are, I think people just have reading comprehension issues. But yeah, I think this, and lot of the objections that are come up, I called them out in Says paper and said, yeah, limitations. I haven't modeled various mechanisms you might do here. I haven't looked at gene sequence. I haven't looked at all the other lines of evidence for common assent. People are basically pointing out to the limitations that I called out in my paper and said, well, you haven't absolutely proven your case yet. I'm like, I know that's not where we're at yet. This is just a baby model.
It seems like there's a different set of standards when it comes to evaluating ID work from any other type of work.
Yeah. So if it were, answering a long-standing evolutionary argument, everyone would be like, oh.
Right. As Silicans in the Q&A asks, the Bayesian statistical methods you apply to tease between these models are pretty complex and not easy to understand in a day. If someone who understood the concepts qualitatively but wasn't a math guru wanted to collaborate with you in testing between these models, how could they
help? Yeah. So one of the things that I would like to do is to try and see, I would like to see come up with different ways of evaluating the models that aren't just the Bayesian model selection, because I think the more ways you can make the case, the better. So if anyone has ideas about how to do that or ways they'd like to pursue it, I'd be happy to hear from them. In that state of I change my mind every 5 minutes about how to pursue things further. So I'm not sure at this point what exactly that looks like.
Again, I'll give folks a few minutes if they want to ask anything else. Otherwise, we could wrap up early. It was really interesting perspective on the issue you proposed that I really look forward to wading through your paper at some point in the near future.
Yes.
Andrew Jones asks, do you think that there might be a way to apply this to biological research, example, medical research?
So I actually had a similar question asked to this when I presented in a smaller group, where someone asked, well, they basically, at some point, instead of worrying about which animals are related to each other, medical people could say, well, which modules are shared, and that could tell you which genes are shared, and that could actually be more useful than trying to do common descent. And someone else in the seminar piped up and said, well, yeah, actually, we already do that. We already care about which genes are shared, not common ancestry for medical purposes anyway. So in some ways, I think it validates, it seems to me, probably how they're actually approaching it already. It seems that I don't think common descent is all that much of a guide to how medical research is done, I suspect that they actually are thinking in modular terms, even if they don't know
it. Daniel Kerr asks, have people tried to generate programs without dependency trees, but purely with descent trees? How is the program qualitatively different or quantitative? That is, can we compare the program expected from these two ways of coding?
So if you're thinking, if someone programmed using common descent instead of a dependency graph. So There are actually some programming paradigms. For example, if you look at Docker, I'm sorry for everyone who doesn't know what I'm talking about, that would be completely meaningless too. That does arguably operate in a tree fashion, but the problem is even there, programmers will copy and paste between programs. So it still doesn't really look quite like a tree. Um, yeah, so I don't know if it's not really a case you could a natural experiment where people do that because as programmers are lazy, they're not going to follow a tree just because you think they should.
Coelacanth asks, 50 years from now, where do you think the scientific community will be in terms of testing the dependency graph versus nested hierarchy in understanding organismal relationships?
It's hard to make predictions, especially about the future. Um, my thinking is in 50 years, we're going to be in a state where those who are on the intelligent design side of things will have either decided that I was a crazy person and no one will remember the dependency graph, or dependency graph will be the standard answer brought out by intelligent design. I think that's the case. Uh, the case being, will intelligent design be able to beat out naturalistic account of it? And I think there's a lot of um, presuppositions and biases in people that determine a lot of that. That I think people don't necessarily decide what they believe on the evidence as much as we probably should or we actually think we do. So I I wouldn't want to make a prediction about how that would play out over the next 50 years.
So, what sort of predictions do you think that we could expect to see over the coming years if indeed your dependency graph model is correct?
Uh, I mean, so I think if it's correct, the model will be successfully extended to sequence or other kinds of data. And there will also be sort of related ideas, I think, from how software is built that will answer other questions about common descent. So I think the prediction, I think, if I'm correct, is that it will further develop into a more full-featured model. And I think if it's wrong, what we're going to find is extending it further just doesn't work.
Ken Ewert says, is it the case that the testing of your model could not have been done a decade or more ago since we did not have the genome classifications that we now have? In other words, Darwin's tree model could not have been falsified without the advance in modern genetics.
um, so certainly there's no way the level of test that I've applied to this could have been done a decade ago. We simply didn't have the genes and all of that classification. It actually, even a decade ago, it would have been hard to come up with the model because with availability of widespread internet access and high-speed internet, actually, in how we share modules in a much more free-flowing way than has ever been the case in the past, it might not have even been possible to come up with the model prior to that point. But certainly, yeah, the data that we didn't have would have made it impossible to do this test a decade ago.
Daniel Kerr says, could my club rerun your simulation? I mean, it is the data set and program open source?
I think I answered this question. The data set is available that I have, well, so the data sets I got are all from, sorry, my train of thought left me. Okay. The data sets are all from publicly available databases you can download. If you go to the biocomplexity website and the supplemental data, you can download my dependency graphs and a script that will score a dependency graph for how well it fits the data. I haven't published my program that actually infers the dependency graph, but if you are sufficiently wanting to do it, my paper does describe the algorithm in, I think, enough detail to actually recreate it if you want to.
And Kevin Ross gives a comment around the question. He says, I find your work very interesting. Thanks for the presentation.
Thanks.
Coelacanth says, cladistics often seeks to build trees, cladograms, by scoring morphological phenotypic traits. Is there any reason that your dependency graph model could not also be applied to morphological phenotypic traits?
So I actually really wanted to apply to morphological traits, but was not able to find a database that sort of had good trait definitions that I could plug into it. Currently, my system works with anything that can be classified in a binary fashion, true, false, present, not. And so if there was a morphological database with true, false values, I could plug it right into the program and see what pops out. I haven't done that largely because I have not found a data set that fulfills the criteria for that at this point.
Anyone else have a question? Or give folks a couple more minutes and then we'll wrap up if there are no more. Okay, Coelacanth asks, how about within biogeography, like when similarly organisms appear in geographically isolated areas, any way to apply your ideas there?
So my knowledge of the biogeographical data is pretty much nothing. So I don't know, and like even broad patterns of what we have in biogeography to be able to assess much of that. And I don't know because my model is based on it defines which genes are in which species or which sort of modules are in which species, and I don't see an obvious connection between which modules you would find in a species and their geographic locations where we'd expect to find them. So I think that there needs to be another model which has an account of biogeography and fossil record and that sort of thing. So it doesn't really fall directly under what's going on in the dependency graph, as far as I can see.
Ken Ewert asks, a tree model explains things better than the null model. What are the other design models that have been advanced to explain the apparent tree pattern? Have they been testable? I think you've already addressed this as well, but carry on.
So we asked specifically about other design models.
Yeah.
There hasn't really been much in terms of other design models. There's some people who've said things like, um, we actually see hierarchies in designed objects, but that's not really a model. Some people have said, well, non-random patterns in terms of how reuse could do it, but again, there's not really a model. The closest you have to a model is message theory's account, which claims that the designer deliberately made life in a hierarchy for sophisticated line of reasoning, which is probably too complicated to get into here. But even there, well, there's not really any testable model to that either, because you're not saying anything different than the standard nested hierarchy claim.
Rob asks, from an apologetics point of view, what does your model provide, if anything, even in its early stage?
So I think for the model from an apologetics side, when someone says nested hierarchy settles the question, it's over, you could say, no, that doesn't settle the question. We have a model that can explain why we see nested hierarchies and show that actually, at least in this early stage, is quantitatively better. And that at least to this point in time, no one has shown some alternate explanation of why there's an appearance of the dependency graph in the data. You are muted again.
Just double check the Facebook live stream and see if anyone submitted anything there. Nope. Okay, so if there's no more questions, then we will wrap up at that point. But thank you, Dr. Ewert, for coming on. It was really fascinating, and I will definitely be dipping into your paper in the next month or two.
All right. Just don't let it interfere with your PhD.
Yes. All right. See you soon.
All right.
Bye, everyone.